Hello, I hope everyone is having a good day. My name is Drew Thornley, and I'll be your moderator today. Welcome to the inaugural PAG webinar series supported by Twist Bioscience. Thank you for taking the time out of your day to join us. Today's topic is cost-effective pan-genome-based platform for genotype imputation, QTL mapping and selection support in plant and animal genomics, presented by Dr. Josh Clevenger with HudsonAlpha Institute. You can submit questions via the Q&A function, and you can ask questions anytime throughout the presentation. The Q&A will take place the last 10 to 15 minutes of the presentation, and any questions not addressed live will be answered via email. Dr. Josh Clevenger is a passionate plant scientist using genomics to develop improved crop varieties around the globe. During his PhD, he played a crucial role in sequencing the peanut genome, a major breakthrough for the peanut industry. After a short USDA NIFA postdoctoral fellowship, Dr. Clevenger worked for Mars Wrigley on the Global Nut Science team, designing and deploying genetic improvement projects across 5 continents where peanuts are grown. Currently, Dr. Clevenger is focused on developing new tools to make crop breeding faster and more affordable for breeders and farmers. His work is helping to create crop varieties that are more resistant to disease and can thrive in challenging environments. Dr. Clevenger, please take it away.
All right. Welcome, everybody, to beautiful HudsonAlpha Institute for Biotechnology here in Huntsville, Alabama. And it's a real honor and privilege to be giving this webinar today for Twist Bioscience in the inaugural PAG webinar series. I want to tell you a story before we get started of why maybe we got here. 4 years ago, I had an inkling of an idea that if we could sequence more samples faster at higher resolution, we might be able to make discoveries that make a real impact for the growers and the people that depend on those growers around the world. And I emailed a gentleman by the name of Paul Dorn, and you might hear at the end of this talk to some extent, and I asked him if he had an idea of how I might do that. And one of the things that Paul did, and this is sort of some of you who are paying attention who might be working for companies, one of the things that Paul did is he didn't try and sell me something, but he asked, Josh, what are you trying to do, and how can I help you? And 4 years later, I'm one of the largest users of Twist FlexPrep NGS library prep, and I'm really excited to talk to you about how we're using NGS and the Twist product to really make an impact around the world. So here at HudsonAlpha, I'm part of my own lab. That's my name, the Clevenger Lab. And what we do is we're really excited about how we can make a real impact. So you heard in my bio that I worked for Mars Wrigley. And in that role, I was really answering to vice presidents who said, if I give you this money for this project, how much return do I get and when do I get it? And it better be within 2 years and it better be a big return. So as an academic scientist, that was a new way of thinking for me because I used to just think about, hey, whenever we make an impact, that's good enough. And now I'm thinking about, well, it has to be as quickly as possible. And here at HudsonAlpha, I get to work with a really talented group of folks. And before I say anything scientific and go into the presentation, I have to sort of point out that most of the work, in fact, probably 99% of the work and the brain power that goes into the things that we do really come from them. And it's a real privilege to work with all of the folks on this slide and to get to interact with them and collaborate with them to make that impact. So if we think about the challenges for genomic breeding, what are the challenges that we face? And basically, the big challenges are how affordable can it be? So you've heard the phrase, it's a numbers game. And essentially, every breeding program, large and small, has to decide how much money do I have to invest in this particular aspect of my program. So to use sort of genomics at scale in various degrees, they have to think about how much am I getting for this investment and how much investment can I really put towards it? It has to be scalable because, again, it's a numbers game. And the more individuals that you can look for, the more wins that you can have. And one of the examples I like to use is that out of the 300 million people that reside in the United States, more so now, there's only one LeBron James. And so out of all of those reproductive events. There was just one example of that exceptional human being. And in the plant world, it's very similar. So you have to have that scale. And then in the genomic space, really, how much can I see? How much variation do I have the ability to identify when I'm looking at my populations? Because again, it's a needle in the haystack, and we're trying to find that one change, that one mutation, whether large or small, that has that major effect for the goal that we're trying to achieve. And then, of course, plant genomes are not simple all the time. Sometimes they are, but a lot of times they're not. They can be very complex. They can be polyploid, meaning they can have multiple copies of themselves or multiple different genomes in the same nucleus. And so that adds an extra layer of complexity. So really, today's talk is about a tool that we use to overcome these problems. We developed Khufu here at HudsonAlpha to really make an impact in the peanut world, a polyploid that has a complex genome. And what we were interested in doing is really developing tools that we could use to accelerate breeding in a more sort of impactful way directly to get cultivars out to the growers. Because what I saw in my work was that when we had a problem, we could put together projects and talk about how we might solve it and how we might get to the end. But I was unsatisfied with how quickly it took us from developing those genomic tools to then deploying them and having a product out the other side. So today, really, I'm going to talk to you about 3 different stories or 3 different vignettes about basically how we're using FlexPrep to scale NGS and discover genomic tools, but then utilize those genomic tools into the accelerated development of cultivars. And the first story I want to talk about is a pathogen in peanut, and this is peanut smut. Now, peanut smut is a soil-borne fungus. In its reproductive cycle, it actually reproduces on the peanut seed. So it infects the developing peanut seed in the soil and then reproduces and essentially utilizes all of the really great things in the peanut that we like to eat. It likes to eat as well. And then when you shell that peanut, it's just a bunch of fungal spores. Now, this disease first reared its ugly head in 1995 in Argentina. And at the time, it's really interesting, they thought someone introduced it for research purposes, didn't pay much attention to it. So didn't do anything to try and mitigate it. And now, in 2025, actually, it's in all of the fields where peanuts are grown in Argentina, and it's caused massive, massive problems. One of the big problems that it causes is its ability to move around the world. So if we look at the potential spread, the teleospores, which are in the peanut seeds, and sometimes you don't even see them until you shell them. So when you harvest the seeds and put them into a shipping container and ship them all around the world, you don't always know which one of those seeds have smut on it. And since that time, where it's become a problem in Argentina, peanut smut has verifiably traveled around the world to different countries, and we think that now it's here in the United States. But until that spread happened, this disease caused major issues for global supply chains. So all sorts of manufacturers and shippers that wanted to take peanuts from Argentina and ship them to other countries and manufacture with them, so process them and put them into candy bars or peanut butter or other products, they had to change the way their shipping routes worked. They had to take them to different places in the countries and burn all the excess after they were done processing them. They had to spend millions of dollars to mitigate the potential risk of the spread of this fungus. So it caused a ton of havoc, and was even Argentine peanuts, which are the largest exporter of peanuts in the world, were banned from many countries. And from the United States, you could not import unshelled, unblanched peanuts from Argentina. So this is a major, major issue. So if we look at having to solve this, where we started was we knew we had a very large problem in Argentina. And really, if you think about competition between countries, the growers in the United States, while they might not have felt like that was a big problem for them, it was a problem with their competition. But for global companies like Mars Wrigley, they were saying, hey, this is a major issue for us, and we need to try and solve it. So they teamed up with the USDA and with the USDA equivalent in Argentina called INTA, and decided to try and develop genomic tools to develop resistant varieties to smut. One of the reasons why you have to do that is because there is no agronomic way to control this disease. It has to be host resistance. So we had to do this fast. We had to do this well. We couldn't take 20 years to identify a gene and then 20 years to develop a cultivar. By that time, the peanut global trade would probably be devastated. So we took an approach, and the field there at the bottom left is the one that we actually mapped in that first year in 2021. We put out all of the early generation individual plants that we had in the population that was segregating for resistance, and we phenotyped those and we sequenced those with what at that time was the iGenomics Riptide. It was an earlier version of highly multiplexable NGS prep. We are able to identify resistance in that first population. And then in a second year, which you're looking at to the right side graph, we are able to validate that resistance in a separate population does reside on that chromosome. We looked at the resistant allele in a diversity population to say, is there a genetic background effect? Meaning, do we actually have the resistance allele, the functional variation for this resistance? We showed quite clearly in 2 different diversity sets that when you had the resistance allele over 3 years, you essentially had 0% smut. And if you had the alternative allele, you had loads and loads and loads of smut infection in Argentina. So moving forward, we took a comparative genomics approach to try and understand what is the exact haplotype for resistance that is giving us resistance to this fungal disease. And we found that in resistant accessions versus susceptible accessions, there is just one area where there's conserved variation that told the difference between them, that sort of separated those 2 groups of individuals. And that was in our resistant locus. And so we identified essentially a haplotype for resistance. And one of the things that's interesting is what we're showing here is on the top is the expression of the genes in developing peanut seeds for the genes in that region. And this is the highest density of resistance genes in the peanut genome. And a lot of those variants that you see there that are indicative of this resistance haplotype are actually in our genes. So although we can't pinpoint the exact sort of functional variation that gives rise to resistance to the smut pathogen, what we can say is this haplotype is a novel resistance haplotype that gives rise to this resistance, and it has high potential to give resistance to other soil-borne pathogens. So really, really impactful discovery here that we're going to be able to test and help solve problems for other diseases as well. So if we move forward, that's sort of the discovery phase. And after the discovery phase, again, we really are mostly interested in getting to that product. So how do we impact the grower and impact that global supply chain? So we actually sequenced thousands and thousands of peanuts, just getting little slivers of the peanut seed from 4 different breeding programs around the world in South America and the United States. And we just sequenced them to identify which ones we could predict as resistant and which ones we would say are susceptible. And what was an interesting thing is the breeder in Argentina that we work with, he's a really funny guy, and he decided he was going to check my work without telling me. So this graph on the left is showing the first brown box plot is our susceptible check. So it's our line that's very susceptible. I'm sorry, this is yeah. Then the next one is showing the resistant check. So that's showing 0% smut infection. Then we're showing you the lines where I said these are susceptible, don't include them in your breeding program, showing loads of infection in this experiment. And then again, the last box plot is showing the ones where we predicted would be resistant to smut, and they had very, very low levels of smut in that test, showing that we could, just by sequencing the seed chips at scale, identify which ones would actually be resistant and utilize that in the breeding program. So on the right side, what I'm showing is a representation of what that looks like. And because we have genome-wide variation, and we have pan-genome graphs that specifically have parents that are localized to the populations of interest, we're able to actually not just select for smut resistance, but other traits as well. So that top line is highlighting actual release of backcross 2 individuals where we could select for smut resistance, resistance to nematodes, and a high oleic peanut. Look at the background with more than 70,000 genome-wide markers, and say this line is most like the agronomically elite parent, and it has integressed these resistance traits. A really powerful way to quickly integress these really valuable traits that we're identifying into elite cultivars. And I'm happy to say that that line is out in the field for seed increase for release right now as we speak. And so in 4 years, a major global problem, we identified a solution and we put in a new variety, and we're getting that out to the growers. So a really magnificent sort of achievement that only is possible because you have this ability to scale genome-wide variation, to scale that WGS. And FlexPrep is really a big part of this. So if we move to sort of the acknowledgements of the folks that had to do with that work, the majority of the funding comes from Mars Wrigley, and some folks in my lab at INTA and the USDA ARS were critical in the success of this project. So now I'm going to take you back, and I'm going to talk more about sort of the nuts and bolts of how this works. So if we think about genotyping, of course, arrays have been used on millions of samples for more than 2 decades, and they're a wonderful tool. Arrays let you look at fixed marker sites. So you're not going to discover anything new if it's not already in your design. That led to folks trying to utilize sequencing. I don't know if that was like an air horn. Utilize sequencing for cheaper. So they would do things like GBS or RADseq or these reduced representation sequencing techniques. Those are really powerful and great, although you have lower numbers of markers at your disposal. So I was really inspired by listening to Ed Buckler as a young graduate student talk about how do we genotype better, faster, cheaper. And I just figured the best way to do that was how do I, as cheap as possible, get whole genome sequencing. And so the best way to do that is to what you would call skim-seq or low coverage sequencing. And that way you have the ability to have access to all the variation in your population, specific for your population. And with the advent of Pan-Genome graphs, which we now have integrated very seamlessly into our operation, you can not only just look at SNPs, but also large structural variants and small indels as well. So really powerful tool, which we're talking about here today. So if we think about the sample inputs, so what are the sort of things that we might use and have used? It's been really fun over the last couple of years. We've actually been able to get DNA and get really great high quality libraries from FlexPrep using snake skin sheds that have just had some lysis buffer and then a bead cleanup, pine needles, hair, of course, and blood. Leaf tissue is the major thing that we get, but also microbiome samples from soil. We like to use seed chips, because with seed chips, you have the ability to actually make a selection before you grow the plant, which is really powerful. But also some fun things like the nail clippings of turtles and lizards as well. And one of the things we're really proud about is really good sequencing libraries from Russian dandelion roots, which have a ton of latex in them. So really high ability to get DNA and get good genome-wide markers from all kinds of different inputs. And if we talk about FlexPrep really quickly, we have this wonderful B-roll of Dr. Kelly Good, who works in my lab. And Dr. Good is just a savant at taking a protocol and really understanding it and making it better and maximizing it. And so you might be interested to know that last quarter, so the last 3 months, she prepped libraries for 120 96 well plates, all by hand. And we do work only 5 days a week and only 40 hours, so we're not actually working overtime. So really an amazing achievement. And the best thing about sort of this normalization by ligation is that you don't have to worry about quantifying your DNA. Once you dial in the DNA extraction and you know pretty much across the board that you're going to have at least a minimum amount of DNA and nothing too huge in some of the samples, you can go straight from extraction into library prep. And that saves a ton of money and energy and time, and it's been really wonderful for our operation. The other thing I really like about FlexPrep is that you're actually doing pools of 12. And why that's important is because before, if I'm doing a full pool of a 96 well plate or even more, If I screw up that library or that pool, then I'm pretty much screwed. I have one sample that's sequenced well or two samples that sequence well and 92 or 93 or 94 that are not sequenced well. Well, with FlexPrep, the pools of 12 really make that a lot better, because if one pool is off, you're still getting good sequencing for the others. So we really like the workflow. We really like the ability of doing large amounts of it by hand, so we don't have to invest in automation. And then that aspect of having really tight control, and as well as if you have a problem, you only have a problem with one of those pools of 12. So really has been a wonderful protocol for our program. And the other thing that I'd like to mention in this next slide here is that you might have seen how Element and Twist have partnered up to use FlexPrep on the Trinity platform for element sequencing. And I wanted to mention this because we did a pilot project, and the Trinity sequencing was incredible. We had a 50,000 probe set where essentially across all the samples there was no dropout, and we had really good coverage for all of the probes that we wanted, which is really important for breeding applications. But the other thing that was great is that the underlying WGS underneath, we were able to call the same amount of SNPs with the Trinity sequencing that we would with a regular sort of NGS prep where we're not getting that enrichment as well. So wanted to make that plug that FlexPrep and Trinity, if you can afford it and utilize it, is going to be a really wonderful tool for breeding applications. If we go back to what Khufu is and what we do, Khufu really is just about maximizing the power of low coverage sequencing. So we really want to have those important markers, that important variation in our population or your population of interest. This slide needs to be updated. We've definitely done more in 65 species, but we're on pace to do 50,000 samples just this year. So it's really just the scale is getting really, really great for all kinds of different breeding programs. And why do we call it Khufu? Well, here's an example of the Khufu pyramid on top of one of the genetic mapping graphs that we use. It's called QTL-seq, and you get a peak where the genetic signal might be. And my collaborator, Wally Karani, is from Cairo, and he grew up down the street from the Khufu pyramids, and he thought that looked just like it, and it does. And so that's where the name come from. So let's look at the process real quick. It's really very simple, but the way that we've worked it out is we took a lot of R&D hours, a lot of time to make this as seamless as possible and as cost effective as possible. So for inputs, for all the things that we do, we like to use seed chips because, again, I can make a selection before I plant the plant. Once I get good DNA, or once I have our input, then we need to have a way to get high throughput DNA extraction. So this is a really difficult step because you want to have pretty normal extraction across your plates. You want to be able to do loads and loads of plates a day and a week, and you want to know that you don't have to quantify every single well, and you feel confident that you're getting as many of those samples, good DNA, as possible. So we currently use the BioEcho Echolution kit. We really like that kit. It's been good in our hands. We, of course, as we've said, now use mostly Twist FlexPrep library preparation kit. We talked a little bit about the pros of that kit. We do that library preparation about 4 to 6 plates a day we're able to do with one technician. And then, of course, we go to low pass sequencing. So we're right now sequencing on the Illumina platform. It's the cheapest sequencing available. You won't be surprised to know that most of the folks we work with want to have the lowest possible price. So we try and basically have that for them, because my goal is not to make money. My goal is to give this power to as many programs as possible. So after we have the sequencing, we go through our sort of Khufu core mapping and filtering. This is our step where we basically have a matrix of all the highest quality potential variants in the population. And that allows us to do all of the different analyses that we want to do, in which include of what I've talked about earlier, sort of trait discovery with QTL mapping and QTL-seq, but also sort of genome-wide genotypes, imputation, diversity analysis, parentage, all the things that you might want to do in your program. And then for the final step of where we like to take it, that marker-assisted selection or genome-wide selection, which is really that how quickly can we take the knowledge that we have and get it into an impactful product to the growers. we have had the privilege to work with a wide array of different plants, and we're just going to quickly show beautiful pictures of those plants and animals and different species that we've worked with. So obviously dahlias get its whole row because what a beautiful flower, but peanut's probably the most important thing there. Loads of flowers, loads of nuts, seeds, and grains. My colleagues are laughing at my joke here. Pumpkins and common beans. If you want to do genetics, work on beans. Beans works wonderfully, but also cacao and hops and millets and soybeans. All these really important things for us. And then we get to the fun slide. That's where we're talking about sort of the turtles and the snakes and the really cute pigs. Most of the time pigs aren't that cute, but sometimes they are. We also do a ton of sort of profiling of fungal pathogens all over in soils and lizards and chickens. Lamb's quarter is actually a plant, but the lamb is a much cuter picture as well, and cows, and of course human beings. And then because it's lunchtime, I'll make you a little hungry depending on where you're at, and these just wonderfully delicious fruits. It's really been a pleasure getting to work on blueberries and tomatoes, and not onions so much, but persimmons and oranges and blackberries and apples and things like that. So really a wide array of genomes, crops, animals, big sizes, small sizes. It all works really, really well. It's been a lot of fun. So we can move on. Oh, B-roll number 2. So we're going to go back and talk about another really exciting vignette of how we were using scaled NGS to discover a really impactful tool. So if we think about the disease, tomato spotted wilt virus disease, it arrived in the United States in the 1990s. So in around sort of 1995, tomato spotted wilt virus disease actually had its peak in terms of yield loss and loss of funds to the industry. After that, there was some tolerant variety and some agronomy introduced, and we were sort of like beating the virus a little bit. But as we know, all viruses bounce back, and recently it's really reared its ugly head and become a big problem again. And there's hope for the future. So in about 25 years ago, there was a line of really sort of not very good agronomic line that was discovered that has really strong resistance. And we knew this for a very long time, and folks had tried to map it for 20 years in various populations and probably millions of dollars and not been able to do it until the advent of scaled NGS and Pan-Genomes. We didn't know what caused the resistance, and now we actually used Pan-Genomics and sequencing of thousands of individuals in a population to identify a really cool duplicated gene cassette of 4 copies of a glutamate receptor that when you have all 4 of those copies, you get very strong resistance. And if we look at sort of how that looks, the other part that's really interested in this as well is that we see segregation of the number of copies in our population. So you can actually have 2 copies or 4 copies, and we've even found 6 copies. And when you have more copies, you have more resistance. When you have less, you have a monoresistant, and when you have 0, you're completely susceptible. So the ability to identify this incredibly impactful tool, and I can tell you, because of the nature of this resistance locus, we hypothesize that it should give broader range resistance to other viruses. So what we've been able to do is actually select lines that have 4 and 6 copies and send those seeds to different countries in Africa to combat a virus there called groundnut rosette virus disease, which has devastated Eastern Africa for 100 years and has just moved to Western Africa. So hopefully, having the ability to identify this tool, select for this tool, and really share it worldwide, we potentially are solving problems that are really impacting people across the globe. So acknowledging the folks that were involved in this, it was a really large effort with USDA, NIFA, and supported also by the United States Senate Committee on Appropriation and folks at the University of Georgia here, as well as USDA ARS. And so the final thing I'd like to talk about is really how this technology is enabling training the scientists of the next generation. So having the ability to say, I can take a big population of peanuts, I can take a sliver of that seed, get DNA, prep that into a sequencing library, sequence it and analyze it, and make some sort of impactful decision based on that really led me to think about how we might integrate that into the world-class education outreach program we have here at HudsonAlpha. And in a collaboration with the city of Dothan and the Wiregrass and us, we developed the Wiregrass Peanut Project, which is really a working breeding program integrated into classrooms in that area. And I have to say that what's been really eye-opening for me is that I'm in high schools that don't even have, they're so rural, they don't even have Wi-Fi. And we're teaching them about how molecular biology can accelerate the improvement of peanuts. And so what we do is we give each student in the class a peanut seed as part of the breeding population. They grow that peanut, they tend to it, they make observations about its early vigor and germination. We teach them about DNA extraction. We extract the DNA of their plant with them. Then we prep with FlexPrep, sequence it. Then they become the breeders. We really give them the information, talk to them about what they might be wanting to select for, and they make the decisions that I bring back to the program and make those crosses. So they're learning about molecular biology, they're learning about agronomy and breeding, and they're learning about how, and a lot of them say this, so I don't have to just say it and be fake about it, they're learning about that there are other things they can do in science other than being a doctor or working in a lab testing for disease, and they get really excited about how they can make an impact around the world. So if we look at what this sort of looks like, it's really quite impactful in the sense that we have plants where we can select for really strong disease resistances and then look at the background and say, this is a plant that I wouldn't have been able to test for all those diseases. I would have missed this. I wouldn't have known the background percentage of the elite parent there, and I wouldn't have been able to make that selection. And we're doing that in the classroom with them. And I am pretty happy to say, and we're in the third year of this program, we now have sort of advanced yield trials of lines that we selected as part of it. So we're able to sort of keep that sort of development going, and then we have this great database of genome-wide variation with individuals that we can mine as we learn more. And that's the best part, because we have all the information, we never stop having the ability to go back and find those that we missed before. So the last thing I want to talk about is about how FlexPrep sort of can work with enrichment. Because it's a more complex library prep, it works really well with enrichment probes. And so we did another study of our 40,000 probe peanut panel, and we're only getting around 5 to 7% missing data across that panel, which is really, really good for for a cost-effective, good library preparation solution. So it's a very, very flexible and useful prep across all the sort of things that you want to get done. And if we sort of finish off the presentation, let's revisit the journey that we've taken today. So basically, we've talked to you about how no matter what your system or what you're trying to do with it in terms of crops that have large genomes or small genomes, or maybe there's just a few of you working on them, or maybe there's loads of them working on them, having the ability to scale NGS in the era of Pan-Genomics gives you a superpower that you probably don't even think that you have. So we go to the next slide. And while today I've talked about examples drawn from my work in peanut, because we don't want to steal the thunder of all the wonderful collaborators that we have, the tools and strategies really go way beyond the single species. We did this to really make an impact across all different kinds of programs, to be adaptable from diploids to hexaploids and even higher, and plants and animals. And we're trying to work with our partners to make an impact around the world. So if we go back to sort of the HudsonAlpha propaganda, we developed this here at HudsonAlpha Institute for Biotechnology. We're a nonprofit research organization. We have more than 15 years of experience advancing genomic science and technology. The Institute is focused on using genomics to improve life with groundbreaking research, educational outreach, and scientific collaboration. And that little sort of illustration of all the dots around the world represent actual countries that we do work with. And so to close, the platform, Khufu platform, is more than just a set of tools. it's really a part of a mission-driven approach to empower you, the researchers and the breeders and educators, with the ability to address your challenge in food security, sustainability, and global health. Because the science that you do is the most important part. The science that I do empowers that for you. And that's really what our goal is. Our goal is for you to do the wonderful things and not have to worry about how you do these other things that we can help you do. And we're really here to help make that impact. And with that, I will close.
Wow, Dr. Clevenger, thank you so much for the insightful presentation. That was dynamic. It was great. I really love those last few slides, especially the global reach. We had a rough count of around 30 different countries represented in the live attendees of this presentation. So I'm sure this was meaningful to all. We will now move on to the question and answer portion of the presentation. Joining us will be, again, Dr. Clevenger and Paul Doran. Paul Doran is Director of Business Development at Twist Bioscience, where he leads efforts around agrigenomics and HTP sequencing. We would like to, again, thank everybody for being here, and let's move on to the question and answer portion. I'm going to start reading these questions to both of you. You can answer amongst yourselves together. I think it'd be nice to have this a dynamic, great, beautiful conversation. I'm going to start with the very first question that came in. This person can say that they were the first person to ask a question on the very first PAG Twist webinar. So thank you very much for this. How is the haplotype defined? Based on what kind of variants?
Yeah, it's a great question. We have different kinds of variants in the haplotype. We have insertions and deletions, and we have SNP variants. So actually, one of our hypotheses, there's a very large deletion in the resistance haplotype that knocks out a susceptibility gene. And we potentially believe that that's what's conferring the resistance. But yeah, I should mention that all the analyses that we do are on Pan-Genome graphs. So when we talk about the variation as a marker, it can be a SNP, an SV, or a small indel.
Great. Thank you. What to do if just 3 to 4 selected accessions are there? I mean, most of the accessions are similar to S-check. To either of you.
So, yeah, Paul, you can try and answer that one. I'm just kidding. No, actually, that's kind of the point of this. And actually, you kind of asked a really good question, because the point is that in traditional breeding, you have so much space out in the field and you don't get to see all the phenotypes, right? You might not have the disease pressure. You might not have the ability to measure. You might not have a drought shelter to measure drought or other things like that. And so the ability to sort of find those diamonds in the rough using the genomic signature is the superpower here. And I go back to my master's thesis where I fine mapped a trait in tomato, and then 8 years later they cloned that gene. I had to screen thousands of tomatoes to find the one tomato that was going to allow me to do that. And so if you don't have that ability, you're either throwing that away or you're just getting lucky. So yeah, I mean, if there's only a couple that you can choose, then that's really the power of utilizing the technology. It's being able to see the unseen.
Great. Paul, anything there?
Well, one thing I'll mention is a lot of people have been doing EMS recently, since there's been non-specific regulations regarding gene editing, and so we're seeing quite a bit of that, and people have been asking Twist to build gene-specific panels so that they can check and see whether or not they've created mutagenesis amongst the genes that they're interested in. So essentially creating more diversity so that they can find that needle in a haystack. So that's something that we can help people out with if they're wanting to move in that direction.
Very neat. Thank you so much. All right. What are ideal parameters to design good SNP arrays for polyploids?
Well, I would say that there's no good parameters to design SNP arrays because I think we should be using NGS. But in terms of if you're asking about sort of like designing or finding the highest quality SNPs, I did do a lot of that in my previous life. And there's a lot of different ways to think about that. And because we're sort of having a dynamic conversation, I'd love to hear Paul Doran's thoughts on the parameters for that.
Yeah, I mean, so the next question is actually kind of a follow-up question in terms of how you might do that, and that's essentially like what is the ideal depth for sequencing on a polyploid. And so, you know, thinking back, so I used to work for Affymetrix for quite some time and was involved in SNP array design. But really the first step was to do the sequencing. And so a lot of it's going to depend on what the population is. You know, if you've got quite a few different individuals, you're going to sequence those at different depths depending on the number. A lot of people will want to take a look at variants that are at least 5% occurring. So, you know, that's like 1 in 20. And so you're going to need to sequence, you know, depending on the number of samples that you have, you're going to want to sequence, you know, at least a couple plates. And hopefully there's good variation within those plates. The depth of sequencing is going to depend on how much variance there is between each one of the individuals and kind of the number of samples. But you know, generally you want to be able to sequence, with a polyploid. That's where it gets a little bit tricky, right? Because you need to not only call it like a standard heterozygosity, you have to do that within multiple genomes. So yeah, so Josh, you can maybe jump in here, but I would say like depth of sequencing for a polyploid. I know when we did a lot of work with wheat, we were doing some of the elite lines at 30x. And then the really, really unique lines, we were just doing reference-based sequencing where you'd use long read. So,
yeah, I would say that sort of Khufu was built on doing this in polyploids. I have a lot of thoughts on sort of how to do that. But what I would say the most important thing is to find the parameters that make your confidence that the read that aligned to that location is aligned in the right location. So having the ability to sort of get rid of the reads that are probably maybe aligned to that location, but maybe not. So you really are trying to find those sort of single locus markers in that sense, because otherwise, in most sort of situations, it's hard to be confident in those SNPs. I will say that we're finding really incredibly, and we haven't dove into this to like really understand how that works exactly. I'm sure there's a really smart reason why. But when you start utilizing pan-genome graphs, a lot of that uncertainty of mapping goes away, because I think what happens a lot is that the sequence context plays a big role. So you've heard about reference bias in terms of reads not mapping to where they should because that reference is too different than the locus where that individual has that individual's genome is coming from. And so when you have the graph, your sequence context actually is more accurate across your population, and those reads are more likely to map to the correct place. And then when you identify a SNP, you can have higher confidence that that actually is a SNP in your population, and it's not just an error that has to do with incorrect mapping because of the context of the locus across the different individuals. And of course, as your system gets more diverse and more complex, that just gets more complex as well.
Great. Thank you so much. All right, on to the next question. Jason from Intertech said, thank you for the presentation. And how do you ensure accurate variant calling and genotype imputation in low coverage sequencing data? And what reference panels or computational pipelines do you use to support this? And I can read that again if you need more. Yeah, go ahead.
That's a great question. If we take it step by step, you know, of course, like there are trade-offs. So one of the things is you lose a little bit of your accuracy, right? Because you're increasing so much the variation that you get to see. And if you're mapping to a single genome or if you're mapping to a pan-genome, that changes. So lower coverage on a pan-genome graph is a lot more accurate than on a single reference genome. So you lose a little bit of your accuracy, but what you gain is the number of variants. Now, how do you ensure that you have sort of like the best possible variants? Well, we have a way the way we do it, and unfortunately it's a trade secret here at HudsonAlpha. But there's a bunch of good ways that you can do that. In terms of how do you check your work? Well, that's the first thing a good scientist should do. So when you develop that pipeline, that you should look at sort of examples where you have a known truth, which we say the known truth is array markers. I don't think it's always true, but it's a nice set that you can use and say, basically, can I replicate the array calls in my low coverage sequencing, impute, and then how accurate are we? And we've done that dozens of times across many different systems. Some are better than others, but we're always much higher than 95% accurate in that sense. The other thing that you can do is just take really high coverage sequencing and then do low coverage sequencing and say how concordant are my calls, of course, and imputation by hiding or masking and saying how good was my prediction of a call that I physically have. And so we've done that a ton as well, and that's another way you can do it. So yes, you're going to lose a little bit of accuracy, but what you gain is the ability to see variation. And I go back to the TSWV example, and I didn't do a good job of saying this, I think, at the time, is that for 20 years, they tried to map that locus, and until we used low coverage sequencing on a pan-genome graph, they were unable to identify the true source of resistance. So I think there's examples across all sort of species of that actually being the case. So I'm happy to sort of reduce my accuracy to improve my power. Paul, do you have I don't know, you probably have some thoughts on that as well.
No, I think you covered it. I think you covered it perfect. I mean, the comparison between the two and then checking how well they match up makes a lot of sense. I do have a question that I get asked all the time when people are moving into a project. And I think with your experience with all the different species that you've shown there, so how do you assess the DNA before moving into library prep? You've got some stuff that's got latex in it. You get different amounts, and in order to kind of move things through your pipeline, I would imagine when you first get something you've never worked on, like what are the steps with that?
Yeah, so it's kind of crazy because I was trained sort of in a really excellent lab of Esther Vandernap, and trained about how to assess the quality of DNA and RNA. And so there's the general ways where you're looking at the ratio of 260 to 230, and you want to see that number around 2, because that's telling you that your DNA is pure and you don't have a lot of other things that have come along that are going to mess with the library preparation. In our case, what we found is if we have too high concentration, that's a problem. And if we have extremely low concentration, it's a problem. So if I'm getting from like a really small amount of tissue, we use 10 milligrams to 20 milligrams of tissue most of the time. If I'm getting like 2 nanograms per microliter to 10 nanograms per microliter, I'm feeling really good because it means that I have enough DNA and I don't have some of those contaminants that are like gonna drive that concentration up. When I start seeing really high concentrations, I get very nervous. And usually a lot of times, those are the samples that end up having trouble. So that's the way we've been assessing it, because we move so quickly. It's really hard to like dial in every new thing. There's always, I was saying to somebody, we had 5 or 6 new species come in just this week with big projects. So we don't at this point have the ability to really dial in every single thing. So it's really about like, that sweet spot of how much concentration you get. You should be very, very suspicious if your concentration is very high when you use a very low amount of tissue. And I have to say, now I know that the exact sort of normalization that you want to have for maybe human exome sequencing or human sort of clinical sequencing you want to have like a much tighter normalization, and so you sort of dial in that purity for the preparation. But for us, the ligation by normalization works extremely well for what we need across very low purities of DNA. It's one of the things that I'm constantly amazed about in fact, we just got a sequencing run I was looking at today, but it was all kinds of different stuff we'd never done before, and it was just this straight line of beautiful normalization. So it really has been super robust for us, and we don't do anything magical.
Awesome, thanks.
Yeah, really neat. Okay, how did you identify a genome duplication using Illumina sequencing and read depth?
Yeah, well, we were able to type it as a marker in the population with the Illumina sequencing. Identification of the variant itself has to come from a pan-genome graph. So we do sort of use short read sequencing on a single reference genome to understand where there are large variants. But of course, if it's larger than the size of the sequencing read, you don't know how large it is. So in that sense, you know something's there, but you don't know what it is. But when you have the pan-genome, of course, it's available to you. Now, the next step is how are you going to see it, right? So if the person who asked this or anyone else in the audience is a seasoned sort of molecular biologist or molecular breeder, you are like, oh, how do I have a really great marker to look at a 63 kb repetitive copy number variant? How would you do that? I actually am thinking that question myself. I'm not sure yet. You can use digital PCR, but that's pretty expensive. You can't really use CASP if there's not a SNP that sort of is isolated within that. So yeah, really the only way to see it at the moment is to be able to utilize sequencing on a pan-genome graph and to utilize sequencing in a cost-effective way on a pan-genome graph. It has to be at low coverage. But we do that. We type that variant in populations quite easily. And because it's a larger variant, it actually gives us more ability to get reads that cover that area. Good question, though.
Yeah, great. Well, thank you. And talking about pan-genome graphs, we've got a lot of questions coming in on pan-genome graphs. So I'm gonna ask this question now. What is the advantage of graph-based pan-genome mapping over reference sequence-based mapping in regards to the detection of structural variants in plant genomic data?
That's a great question. And the advantage really is there's a couple advantages that pop to mind. The first thing is computationally, especially at scale, these analyses are expensive, right? Even if you're looking at low coverage, it's computationally expensive to analyze thousands of individuals, mapping, calling variants, and all those things. One of the things that I should have known, but I didn't think about, is that the ability for the mapper to identify where the read goes on a pan-genome graph is vastly superior, because most of the computational time and power is the mapper trying to understand where do those reads go, where they're in repetitive regions where it's like, I don't know where it's to go, and it has to do all sorts of extra things to make a decision of where that read aligns. And so when you sort of take that away, because you sort of lengthen out and you smooth out that context and you have that sequence context across your population in a much sort of more accurate way, the computational speed and power, you can do it faster for less computational power. So that saves you money, right? Because compute costs money, and that's really important. It's not cheap. So that actually is a big pro advantage. And the other advantage is, of course, is I can take a 150 base pair read, have it align into a massive structural variant and say, this structural variant is present in my population. And that's just, in a single reference. I can give you an example. Peanut has very low diversity. We expect, and you can look at the peanut genome papers in Nature Genetics, and we've done some of this work. I can expect one SNP per 10,000 base pairs between any two cultivated peanuts. That is extremely low diversity. But in our 18 parent pan-genome graph that we use in our population, we've identified indels within 1 kb of genes, 50,000 of them, and these are indels that are 200 to 5,000 base pairs large. That is an extreme number of potentially impactful variants. On one of those peanuts as a reference, I would never be able to see. So the advantage is incredibly massive. It's almost, really hard to quantify.
Great, thank you. All right, and moving on now, that rolls right into how cost effective is this compared to GBS in a diploid species such as maize?
Yeah, that's a good question.
Yeah.
I don't think that people calculate cost correctly, quite frankly, because if I spend double the price on my experiment, but I get an answer in year 1, versus I use GBS, which might be cheaper, and then I get an answer in year 3. I've now incurred FTE costs. I've incurred farm costs and phenotyping costs and greenhouse costs. I've paid for all sorts of things I wouldn't have had to pay for. And there's the cost that I don't have an answer to maybe a potentially extremely impactful problem. I mean, we're talking about diseases that are costing hundreds of millions of dollars across industries. So with all that being said, I do hear about how much folks pay for GBS in a lot of commodities. Now, maize might be a lot cheaper because there's a lot more of you, but what we do is still cheaper. So it's actually, even though you saved all that other money that you didn't put into your cost, it actually still is cheaper, and it still is cheaper than a lot of arrays. So not across the board, but the majority of them, it still is more cost effective. So it really is, kind of a no-brainer.
Yeah, I would concur with that. I'd also say that over the last 10 years, sequencing prices have dropped. In the last 2 years, they've plummeted. And so the whole idea of doing reduced representation with GBS was to make it so you could afford to sequence what you were sequencing. And now with sequencing costs and the availability of sequencing in different platforms, it's no longer a cost thing. Now it's just a matter of know-how, and people that know how to do either low coverage or some sort of targeted approach after you've done a low pass approach, understand that the cost differences are really don't exist anymore. In some cases, low pass is even less than GBS.
Yeah, agreed. And they still want lower cost on sequencing. So who knows where it'll go, right? All right. So is this technology capable of detecting if there is gene disruption by transposons?
Yeah, that's a great question. I'd love to find out. I think so. We haven't done that. Have you seen anyone do that, Paul?
I guess not necessarily. I guess if you were to do whole genome sequencing at a little bit deeper coverage, then you would probably be able to map those together and see where the new intersection of the disruption occurred.
Yeah, we identify variants that are variants caused by disruption by transposons within our graphs, right? So right. If you're talking about like, this is the individual that has the phenotype of interest that is controlled by some disruption, I have that genome in my graph. Yes, you absolutely can see it. On a single reference, I think that's a different question.
Yeah, I think the beauty of sequencing is that, if you have a population where there was a gene disruption and that became something that you selected for because it created something either good or bad, it would keep showing up, and it might show up in unmapped reads. And if you've got a whole bunch of unmapped reads and then you see that there's a common sequence in there, you could probably
Okay, I think I might have locked up a little bit. Can you still hear me?
I can hear you, yes.
Okay, good. For some reason, I thought I lost all sound there, but here we go. All right, we're on to our last question here. I know it's getting late in the timing. We have our audience is staying with us tight, though. We still have a lot of people on here, so let's get to this last question and then wrap it up. What approach do you use to align short reads to the Pan-Genome graph? What approach do you use? Do you take the approach such as Pan-Genie?
So he's talking about do you take a Kamer approach such as Pan-Genie?
There you go.
Yeah, we don't use Pan-Genie. We use VG tools, and we use VG tools to align, and then we manually call the alleles from there. But there's a lot of really good tools available to do that, and I would be remiss if I didn't say, like, just incredibly thankful and impressed by those folks that build those powerhouse tools that we're able to then make this difference with. But yeah, we use VG tools.
Great. All right, Paul, anything on that?
No, thank you for everybody attending. Thank you, Josh. And it's really a privilege to be part of the first PAG webinar series.
Yes, thank you all. That concludes this question and answer. Unless you guys, we still have a lot of people on. If you guys want to talk a little bit or have another couple of questions on your own, otherwise I will wrap this up.
I guess I could ask Josh one more question.
Yeah.
Josh, what's the largest genome that you guys have worked with?
Well, we have worked with Onion, which is 16, right? We've worked with Silphium, which is 8, and we have built a phased loblolly pine genome that we do work on. So that's 22 gigs. And for the loblolly, actually, we do great with getting, which is really fun for me because I work in peanut, which is a 2.5 gig genome. So when I get 1x peanut, it's 0.1x loblolly. So just doing sort of like general analysis on 0.1x, it works really well. But we also have a really nice, and I should plug Twist for this, a really nice panel, which basically just enriches the genes in the genome. So we're just sequencing sort of the gene sequences of loblolly, and that works quite well as well. And I will say that Paul mentioned EMS populations, and we recently did a big study with 1,000 individuals, and I was really, really blown away by the results because we used a gene-specific Twist panel, and it really did essentially enrich for most of the genes that we were interested in, and all of the potential EMS mutations were in genes. And we also did sort of genome-wide sequencing, and that's where it was super cool because we saw that, like, outside of genes, there was essentially, in the enrichment, there's essentially no variation, but there was variation in the whole genome, meaning that we really did a good job of enriching for the genes. So I'm really excited about this idea of enrichment panels that are just targeting whole gene sets, because I think that's super cool.
Awesome.
Yeah, thanks,
awesome.
Paul.
Thank you, guys. Yeah, that was so good. Thank you so much. Wrapping it all up, we would, again, like to thank Twist Biosciences for their support of the inaugural PAG webinar series. Thank all of you for joining us today. Again, you can all say that you were part of PAG's history being in the first webinar that we produced. Woohoo! So everybody, if we didn't get to your question, we will answer it via email. You'll get an email from Twist or from Josh answering those questions. Please look for the post-webinar survey via email. Your feedback is very important. We want to know how we can do better, what you're interested in. If you have ideas for other webinars, please let us know in that survey. The on-demand webinar will also be available on the PAG website within 48 hours. So if you want to go back and look at any part of it, you can. We look forward to your attendance of future PAG webinars. And we also look forward to seeing you guys in January in San Diego. So have a nice day. Thanks, everyone.
Thanks, everybody.
Mm-hmm.